Okay, here we go. Yeah, I'm on slide 47. Okay. So here we go, guys. Let's go ahead then and look at the
that's a feeling.
So we're gonna take this paper. Here we go, class. Hey, now. So let's go ahead and solve. This is something similar to what College Board would give you, the same types of logic and thought and stuff. So we're looking at a space probe discovers a planet inhabited by creatures that reproduce with the same hereditary patterns seen in humans. 3 of the phenotypic characteristics of these creatures are height. So again, when you were like reading stuff about genetics, I tend to always write everything down. So we have height, we have antenna, or we have like nose, if it's upturned or downturned noses. And so you guys just grab that paper. We're working on the genetics paper on the front of my desk. And so here, Earth scientists were able to do some controlled breeding experiments with these organisms. 100 males and 100 females were used in experiments, and the results of the number of crosses are shown below. Review this information and use it, let's just say, to answer the questions that follow. So in cross number 1, we have true breeding homozygous tall creatures were crossed with true breeding short creatures. So right now, we don't know if tall is dominant or short is dominant. All we know is it's true breeding, which implies that they're going to be heterozygous. So here, we see in the offspring the numbers. So we see for tall, the males and females are about equal. That's going to imply so far that doesn't have anything to do with like male or femaleness, like it's not a sex-linked trait. And then we see it's not exactly a 3-to-1 ratio, but it's also not 50-50. So these numbers, I did the math, it's really not that close to 3-to-1, but we're going to go with, it's a 3-to-1 ratio for the F1 offspring. So that means that with true breeding, if you have, let's just use like alleles, in true breeding, that means they're homozygous. All of the F1 are going to be heterozygous. But here, we need to look at the data to determine what is dominant and what is recessive. And so if we look at a 3-to-1 ratio of the F2, so when we get to the F2, this is what's been crossed. So we cross an F1 and an F1, and then you get 3-to-1 ratio here. Oh my gosh, this marker is not really the best choice. But you get your 3-to-1 ratio. So now we can make inferences that the tall, is what's dominant and the short is what's recessive. Because we see a 3-to-1 ratio of 3 tall to 1 short. So this is not sex-linked and it is autosomal. Height is tall would be inherited autosomal dominant, short would be inherited autosomal recessive. Now we look at the next cross, and it's dealing with antenna. So we have true breeding individuals that have antenna. I'm just going to write ant, are crossed with no antenna. And all of the F1 have antenna. So the F1, they all have antenna. And then the F1 creatures were crossed, and the data was obtained. So when you cross 2 F1s, you get, again, approximately a 3-to-1 ratio. And there's not really any difference between the males and the females. You can see here the males and females are about equal. And the without antenna are also about equal between males and females in the F2. So that's also going to tell us, okay, antenna are going to be dominant and no antenna is recessive. So when I did this cross of the F1, I was crossing 2 heterozygotes together. And you see the 3-to-1 ratio of antenna to no antenna, right? And so it's by looking at data and ratios that you can determine what is dominant and what is recessive. Oftentimes, when you cross 2 true breeding individuals, your F1, so in this situation, you had an antenna crossed with a no antenna, and all of the F1 had antenna. That tells you antenna is dominant. to no antenna, because that F1 is heterozygous. And so if you cross two heterozygotes, you can kind of confirm this when you see a 3-to-1 ratio with antenna and no antenna. So that recessive trait is masked in the F1. So when people in the general population say, oh, it skips a generation, like that's what they're talking about. It was in like the grandparents and now it's in the grandchildren. Okay, it skipped the parents. Whatever. I hate that. Okay. Next, cross number 3. All creatures that are true breeding for upturned snouts. So now we're on number 3. You have upturned snouts. True breeding are crossed with creatures with downturned snouts. All of the F1 had upturned. So the F1 are all turned snouts. Their noses point up. And so the F1 creatures were crossed. So then you have an upturned snout crossed with an upturned snout. And you get both upturned and downturned. But what do you see in the data? Is there evenness between males and females? No. So here you can see in the F2 that there are no females with downturned snouts. So let's go ahead and do a cross with this. So let's go ahead and just see if we can cross this. So here we have an upturned snout. Now, all of the offspring have an upturned snout. Who do you think in the F1? In the F1? Who do you think is the parent that is upturned snout to begin with? The male or the female parent? It's going to be the female parent. Because if you think about it, could it be the male parent? Yeah. Because if it had been, let's use, what letter should we use? N? For nose. There you go. Okay. So if we did this, let's say that N is upturned, is dominant. We know that is the F1. And lowercase n is a downturned snout. If this mom had been this true breeding downturned snout, then in the offspring, in the F1, half of the males, or like half of the males would all have been downturned snouts. That did not happen. We have all of the offspring have upturned snouts. So that tells us that the mother in the parental generation was homozygous dominant for upturned snout, and it was the dad that was the downturned snout. So then here, we're going to do this, where you have the cross, the original parental cross. So when we do a Punnett square for the F1, oh no. So here we have those females are heterozygous. And then for the males, like this. Can you see that over there? So now, let me get a more fine tip pen real fast and see if that makes life easier. So now, then it says that an F1, the F1s were crossed. So I'm going to take a female here and a male here, and we're going to cross them. So to make the F2, here is my parental cross. Here's the female. She's heterozygous, crossed with the male, like this. And so when I do my Punnett square, I can see here's the female. These are the possible gametes due to Mendel's law of independent segregation. Sorry. So when we look at the data, we can see in the F2 generation, there are 0 females with downturned snouts. And that's true. All of our females have upturned snouts. And in the males, it's 50-50. Half have upturned snouts and half have downturned snouts. So this is an example of upturned snouts would be inherited autosomal, sex-linked dominant, and downturned snouts would be inherited sex-linked recessive. Okay. How you guys doing so far? But of course, the teacher makes it look a little bit easier, huh? All right. Next, we have the last one, cross 4. Says true breeding tall. Okay. True breeding tall with antenna are crossed with true breeding short without antenna. And all of the offspring were tall with antenna. Okay. So I wasn't sure when reading the question what the genotype was for this antenna, because all it said was true breeding tall with antenna. You can like make assumptions sometimes that when they say true breeding, they mean true breeding for both, but I wasn't sure. So I just made a little line because I only know that for sure they have one allele. I don't know what their second allele is. But right here, they told me that the F1 all were tall with antenna. So here, all of the offspring in the F1 are tall with antenna. So that tells me that this parent was also true breeding for antenna, because if it wasn't, it would be a 1-to-1 ratio. If this parent had been this, when they crossed, I would have had half, if I did a Punnett Square, half of the F1 would have been antenna, half would have been without antenna. So that in the F1, they say all do. Okay, I know it's homozygous dominant, 100%. Is that okay? Yeah. So now here, the F1 were crossed with a true breeding short without antenna. What's another word for that? Test crossed? Sorry, test crossed. So then here you have your F1 were test crossed. Oh, I did that backwards. Does that matter? Okay. So they were test crossed. And the F2 data is in the table below. What would be our expected if these traits were inherited on different chromosomes and they were independently inherited? Yeah, you'd expect a 1-to-1-to-1-to-1 ratio. This is what I did on the board earlier, but in case you came after the break, let's go ahead and just show this. So here we have different options. So this individual here is heterozygous for the height. These represent like the chromosomes, by the way. So then here's the other chromosomes. And for antenna. So in Mendel's law of segregation, the alleles separate. So if these separate right here in gamete formation, one gamete will have this and the other gamete will get that. However, you could have independent assortment, and this is possible. That can be reversed. So therefore, you could have a dominant for height, tall, with a recessive for no antenna, or you could have a short be inherited with an antenna. So this is based on Mendel's laws of segregation and independent assortment. Here are the different possibilities of gametes that could form from this parent. So here, when I set up my dihybrid cross, my large Punnett square, I'm going to just put those parents at the top here. And then the other parent, though, because it's test crossed, all of their gametes are going to look like this. That's like all they have. So then this gamete will go here. So now when I do this, I should end up with a 1-to-1-to-1-to-1 ratio. Okay? And so if this was actually a like a chi-squared problem, and they wanted to know, because our data is showing us not equal. It's not 1-to-1-to-1-to-1. They're not all evenly inherited. So we would need to add up. So can someone take their calculator on their phone and add up our total number of offspring? Is it 10? Does it say 10,000? No. It just does? Okay. So if you add up all the possibilities, there's 10,000 offspring. So our expected would be 2,500, 2,000. How funny. I used the same numbers earlier. And then you would use a chi-squared. And you would reject null hypothesis that these traits are inherited independently. These numbers that we get here, what we're seeing is that these offspring look like neither parent. Those are due to crossing over on in this parent. They're due to them being inherited together. So that heterozygous parent, guys, when we talk about linked genes, this is how it turns out. If here's a chromosome and here's a chromosome. It could be inherited like this. They're heterozygous for both. But then you can have crossing over. Crossing over would happen like here. and now these A's get switched. So that's kind of what's happening. Like those are the effects of crossing over, because now crossing over switched them, not independent assortment. Okay, so that's what's happening. All right. So there's just an example. Whoever wrote this question modeled it after an FRQ from College Board. So these are like very similar to the types of logic that you'll need to need. So at this point, it's 10:35. I have less than an hour and a half left. And in real life, my kid has a soccer game at noon, so I don't really want to hang out and go past 12 o'clock. And so we have a lot to cover, so now I'm going to kind of speed things up. Is that okay with you? Okay.
You hold it. Read it out loud. So this is what.
Yeah. You guys, can you make the sign-in sheet still go around? I don't think this side of the room did it. So we left off at like pedigrees was the last slide in my PowerPoint, and Mendel's laws of inheritance and stuff. So now there can be errors. Oh, I really am going to speed things up. Okay, here we go. So in Mendel's, or not Mendel's law, but sometimes there can be errors in meiosis, and there's two different types of non-disjunction. You have the if the homologous chromosomes fail to separate, if that happens, then every single gamete has an incorrect number of chromosomes, or sometimes just sister chromatids can fail to separate. And there you have two gametes that turned out okay, and then these ones have an incorrect number. So this can lead to like Down syndrome has an X number 21. And so there's different genetic outcomes to this. Klinefelter's, Jacob syndromes, Turner syndrome all come from incorrect numbers of sex chromosomes. Okay, many ethical, social, and medical issues surround human genetic disorders. Yeah, I don't know how to help you with that standard. Like, I don't. Guess what? What do I mean? Good luck. I don't know.
Nothing on there?
Many traits are the product of multiple genes and/or physiological processes. So we already talked about polygenic traits. So your skin color is an example of polygenic. So if you really wanted to do a Punnett Square, like two hybrids. So a hybrid are like heterozygous for all three traits. If you wanted to cross them, look at the possibilities of their offspring. Like here, two people, but like medium skin color, I guess, heterozygous for all three of the genes involved. They could have this offspring and they could have this offspring. Those are both possible. Okay, like more likely though, you're gonna have something like this. But anyway, so some traits are determined by genes on sex chromosomes. We've already talked about like hemophilia and autosomal or autosomal, a sex-linked recessive or sex-linked dominant. Males get only one copy of an X chromosome. So a lot of times when we talk about sex-linked, we're talking about X-linked because the Y chromosome only has about 30 genes. And the X chromosome has about 1,000 that don't all deal with sex. Here, like hemophilia is an example of a sex-linked inherited disease. So carrier would be fine because they have one producing X, but the males either have it or they don't. Now, sometimes word problems call heterozygotes carriers. If you're a carrier, it means you carry the allele with you, but you don't express it. Okay. So here you can just follow that through with this is with colorblindness. So males often will be more males in the population will be colorblind than the females. If a female is colorblind, all of her sons will be colorblind because all of the sons get their X's from their mother. Okay. And some traits are non-nuclear. So earlier on the pedigrees, when I talked about mitochondrial DNA, like what would this look like if it was a mitochondrial mutation? Well, you get your mitochondria from your mom. So we can trace mitochondrial DNA through the maternal lineage. So I have two sons. My sons have my mitochondria, but it ends there. My mitochondria will never be going on ever again because I am not having any more kids. So even if I have granddaughters, it's not my mitochondrial DNA. It's from their mother and their grandmother on whoever my sons mate with. But I do have girl cousins who may have daughters. Will we share the same mitochondrial DNA? Because my mom, it's my aunt, my mom's sister. So we have my grandma who put mitochondrial DNA into my aunt and my mom. And then my aunt had 3 daughters. They don't have kids yet, but if they have kids and they have daughters, then mitochondrial DNA will be there and it'll be the same one I have. So it's not as sad as it sounds, right? And so, and then Y chromosomes are inherited through the paternal line. And so I don't know how accurate this statement I'm going to make is, but I vaguely remember reading somewhere, but it could have been 10 years ago, and I could be remembering it wrong, that there was an emperor in China, however many generations ago, that had lots of wives. And today, about a fourth of the men in that region have that Y chromosome from some emperor from like 1,000 years ago. So the Y chromosome stays from father to son, just like the mitochondria go from mothers to daughters. Okay. Then also the mitochondrial DNA, because mitochondria evolved from prokaryotes that have plasmids. We're talking about plasmid DNA in the mitochondria. Also, you have plants. When they have chloroplasts, the chloroplasts also have their own plasmids as well. So, all right. Ooh, a break. Regulation time, guys. And we look at operons. So both DNA regulatory sequences, regulatory genes, and small regulatory RNAs are involved in gene expression. So let's go ahead and look at operons first. So this is in prokaryotes. This is not in us. We are eukaryotes. We do not have operons. So when we look at this, an operon is a promoter, operator, and all the genes they control, probably for some kind of metabolic pathway. If we talk about all the genes they control, well, maybe each gene codes for a particular enzyme that's used in a pathway. So it's beneficial and has been selected for to be able to express them all at once. So with this, here, the bacteria, the prokaryotes, are going to have a regulatory gene. That's not part of the operon. The regulatory gene is coding for the mRNA that then is translated into a protein. That's going to cause a protein that will be the repressor. And if the repressor is attached to the operator, that'll block RNA polymerase from transcribing. And those genes, that operon would be considered to be off. So we look at the trip operon here. There's the regulatory gene that is coding for the mRNA that codes for the protein. Well, in this operon, this is coding for the enzymes that will be used in the metabolic pathway for the bacteria to make tryptophan. Tryptophan is an amino acid. So as the bacteria makes tryptophan, eventually that tryptophan may accumulate to such high levels, they're like, dude, we have enough. And it needs to shut down that operon and turn off the production of tryptophan. There's two ways. You can do feedback inhibition, but you could also do this tryptophan can also act as a co-repressor. And that co-repressor will then attach to the repressor protein, changing its shape, making it fit into the operator, attached to the operator, and now transcription is blocked. And now those enzymes aren't made and no tryptophan is produced. But it's like a temporary thing. It's like kind of, if you really want to think about it, kind of like homeostasis. Keeping tryptophan levels balanced inside the bacteria, but not really homeostasis in the same sense that we do. But that's how I always think about it. Okay. Do you want to, can you flip the screen so it's like up more? As weird as that sounds? Like you just look in my head. There you go. Okay. And so then we have here inside our intestines, we have E. coli or lots of bacteria that live inside of us. So it depends on what their environment is. So this is how bacteria or prokaryotes can respond to change in their environment and change in metabolism. So when you eat dairy, for example, dairy has lactose. Lactose is a disaccharide that the bacteria can use as a source of energy. So what will happen when lactose becomes available, they want to take advantage of it. So they're going to produce enzymes that will then do hydrolysis and break those glycosidic linkages. So now that bacterium can then use that energy or use that sugar to make ATP. So how do they do this, right? So now this operon is an operon that's going to be normally off. This is an operon that's called an inducible operon. It'll be induced or a molecule that will attach the repressor, moving the repressor off of the operator. And now RNA polymerase can be transcribed. It'll attach the promoter sequence, transcribe, make the messenger RNA. That will then code for the enzymes necessary to break down lactose. Okay. So in the bacteria, they have two different kinds of operons. They have a repressible operon and an inducible operon. Lactose, the lac operon, is inducible, meaning that it's usually in the off position. So when that protein is first made, that regulatory protein, it fits onto the operator just fine. But now when the lactose becomes available, lactose will act as an inducer, attaching allosterically to that repressor protein, changing its shape. It's like a conformational change in shape. So now it will not attach to the operator, and RNA polymerase can transcribe and make the mRNA and then the the proteins necessary to make the enzymes to break down lactose. Okay. Ooh, both DNA. Oh, so there's also like Regulatory RNAs, that's where you're going to use your microRNAs. So microRNAs are kind of rad. So if here you have a ribosome that's going to read the mRNA to produce a protein, a microRNA will actually attach to the RNA, blocking the ribosome and stopping protein synthesis. So when we talk about regulating gene expression, like in eukaryotes, we have transcription factors, we have chromatin that can be like heterochromatin or euchromatin. But really, if we've made it so far to make the protein, we can also sometimes like stop the protein from being made, and that's where we're going to use microRNAs. So they'll block translation by base pairing because it's RNA. So when we look at this, here you're going to have the you're going to have, like, if this is the RNA, like A, C, G, U, I don't know, C, C, A, U, like that. So the microRNA will then base pair with this RNA. So then this would be the microRNA coming and attaching. So it still uses base pairing rules. Okay. And then we have transcription factors in S. So with S, if you remember, we have our promoter region, and a promoter region in the upstream of the DNA. So if this is opened up, you have your enhancer region. And your enhancer region is where very specific transcription factors are going to attach. So you have specific transcription factors and you have general transcription factors. And they're all going to form a complex called the transcription initiation complex. And what's going to go down is they're going to bring everything together and aid of some kind to bring about a cell response. So here, we talk about like turning on gene expression or not. A lot of times when signals attach to receptors, a lot of times it could be turning on gene expression to promote whatever. Like here's a division promoting gene to have the cell divide. Sometimes though, messages can cross right through the cell membrane. If it's a steroid-based or lipid-based message, it can cross right through the cell membrane as well as the nuclear envelope, attaching to an intracellular protein, and together that'll turn on gene expression. So the proteins that drive growth and division. Ta-da! So great. Okay. We can regulate gene expression at the chromosomal level, where here we have either heterochromatin or euchromatin. And if you remember, heterochromatin is the more condensed chromatin that makes it harder for RNA polymerase to attach to. Then you have euchromatin, which is more loosely bound and opened up, where RNA polymerase can attach and do transcription. So generally, genes within heterochromatin are not expressed. And then you can modify, though, the DNA or the histone proteins to then create this type of chromatin, whether it's eu or heterochromatin. So when we look at this, you can add acetyl groups to the histone proteins. When that happens, that's going to, poor RNA polymerase can't transcribe. But when you add acetyl groups, now what's going to happen is the chromatin is going to unwind. Now RNA polymerase can come and transcribe and then make the messenger RNA that makes the protein, and the gene is expressed. Oh, but if you take off these acetyl groups, it goes back to heterochromatin, sorry. And now transcription is blocked again. So acetylation is like a more of a temporary alteration of chromatin structure to either promote or block transcription. Then, however, you have methylation. Now, this is different because in methylation, the methyl groups will actually modify your nucleotides. They attach to your bases, and they actually add to the DNA. And these methyl groups will actually cause it to be heterochromatin, which blocks gene expression. This is a more permanent alteration to the DNA. It can be reversible, but acetylation is more of like a temporary, like fluctuations and stuff. Okay. So here in DNA methylation, this though is how you. This standard actually says transcription factors. You can have different life experiences, different nutrition levels. So nutrients can also play a role in gene expression, and you're going to have outside factors influencing the expression of your traits, and that deals with like epigenetics and that kind of stuff. Okay. So here you can see whether transcription is happening or not. So that would be like with different regulations, even though they have the exact same DNA. Thank you. And so here, but you also have environmental factors that can deal with regulation. So you have exercise can produce certain signaling molecules. Nutritional factors have an impact on your gene expression, and emotional health, as far as like meditation and relaxation, because your stress levels can have an impact on things, as well as environment. So clean air, water, and soil. Like when people breathe in secondhand smoke or air pollution, that actually is methylating DNA and increasing your chances of asthma. And so if you live in a high air pollution area, your chance of asthma are increased. Now, if you add that to secondhand smoke in your household, your like chances of asthma are like so high, all because of the way the environment is affecting gene expression. This is also why it's so important to eat healthy. Eat healthy, exercise. It's not just like, oh, for looking good, but for your gene expression, and you're lowering your chances of like cancer and other health-related issues. Okay. Signal transmission within and between cells mediates gene expression. So when we talk about like signal transmission between and within cells, this is how cells communicate. So cell communication, when they send out ligands, they attach to like receptor proteins, activates a signal transduction pathway, and it turns on gene expression, or possibly turns off gene expression. So that's what this center is just pointing out to you. Like gene expression is going to be based on communication between cells. So this is kind of like when we talked about like platelet-derived growth factor, when the platelets receive like a message, and then what happens is you have, and then the cells receive the message, oh, time to divide and repair that cut. Or you can talk about when you have like the helper T cell. The helper T cell sends out cytokines to the killer T cell. The killer T cell receives that message, activates a signal transduction pathway, are going to attach to a receptor. What kind of molecule is this? Is this protein or lipid-based? Protein-based or water-soluble because it can't cross the membrane. It's going to attach to a receptor on the surface. And then you have relay proteins, or like a signal transduction pathway within the cell. This can rely on like kinases and stuff. It'll amplify the signal inside. It also relies on a second messenger. The second messenger we used in class was cyclic AMP, but there's other ones. So if you see anything about a cyclic second messenger, like in questions, they all kind of have the same purpose. Okay, even if it's one we haven't learned about in class. You have your kinases, and you have a transcription factor that will influence gene expression. This signaling molecule is what kind? Lipid-based, because it's crossing through that lipid bilayer and attaching to a receptor inside, and will directly alter gene expression. Okay. So here, I think just how I mentioned earlier about the immune system. So you can see how chemicals being released will turn on gene expression for different things. Okay. Here you can see how signal molecules go real close. Okay. About neurotransmitters or something? And said, explain what's happening in each of the 3 steps. So a 3-point question, you get 1 point for this part, 1 point for that part, 1 point for that part. But what? Signal transmission within and between cells mediate cell function. So with this same thing, cells communicate and can influence their function of what they're doing, what they're producing. I don't know. So like if you talk about signal transmission of what their function is, so when you talk about glycogen in the liver, when you release like epinephrine, and epinephrine attaches to the receptor on the surface, epinephrine is part of that fight or flight response. So if you get scared or something, then what's going to happen is you're going to have your adrenaline or epinephrine to be released into your body. When it attaches to that G protein, it's going to activate a signal transduction pathway using the second messenger. And at the end of that, like inside the pathway, the signal transduction pathway, you'll have the enzyme is going to be activated that then causes glycogen to be broken down and released into your bloodstream to increase your blood sugar, so that way you can like fight off or flight, like run away or fly away from the predator or whatever's scaring you. Oh, like hey, the morning of your AP test, let's stay calm and like not let this happen. Okay, you're gonna be nice and balanced and like ready. Okay. All right, so here, just another way. Oh, this is I think using with light as a message. I kind of like this one here. And so light can also activate a signal transduction pathway. So this would be in like plants. Ooh, they have phytochromes. They have light receptors that can turn on gene expression. Okay. Here, I like this because it shows about like DNA damage and stress. There's different things that can happen in response to that. So sometimes like a cell can receive signals that say, hey, stop. There's DNA damage that can either lead to the cell not going through the cell checkpoints. Oh, it can go through apoptosis. Maybe they could try and repair the DNA, etc. So another way to look at that standard. You have homeotic genes or Hox genes. So with this, signal transmission within and between cells mediate cell function. So when we talk about like development and which genes I like that. Like the Hox genes are giving directions of what to form. So Hox genes are dealing with like anatomical structures, and they turn on like if we look at like the body plan of this fly, there's different Hox genes being expressed in different segments that will have different structures form in the correct places. Scientists have fun with these little flies, and they like turn on Hox genes for legs. They've grown like flies with legs at their foreheads, or flies with two sets of flies with like 2 sets of wings. So it all depends on which Hox genes are being turned on or off. Okay. And then here's the generic signaling pathway. It can lead to altered metabolism, altered gene expression, or altered cell shape or motility. Okay, alterations in DNA sequence can lead to changes in the type or amount of the protein produced. And the consequent phenotype. Hey, let's talk about mutations. Mutations can change things. So when we talk about mutations, it's not a mutation in the RNA. So if you talk about like, oh, here's some DNA, and this DNA, let's say you're making RNA from it in transcription, we're not talking about just one-time error in RNA. We're talking about when DNA replicates. When the DNA was double helix and it like opened up and you used one strand and the other strand to build the complementary base pairs, and there's errors. So now the daughter strands are going to have like mistakes in every cell generation after that. So mutations happen in the DNA, and then that is transcribed into RNA. And now that RNA is read in codons in groups of 3, you may get different amino acids. So with that, here, sometimes if this is like the normal like amino acids, the polypeptide forming, sometimes that mutation can create a stop codon, and now that protein stops too early and it's not produced. I don't know, that may, remember we talked about Tay-Sachs? We can pretend that's what happened in Tay-Sachs. I don't really remember exactly, but that would make sense to have a stop mutation, and now, or a nonsense mutation, create a stop codon, and now there's no enzyme to break down fat in the brain. Here, the genetic code. The genetic code is redundant, which kind of helps us out in terms of mutations a little bit. So we look at like the amino acid isoleucine. So isoleucine, if you have the DNA, the DNA that codes for AUU would be TAA. And if TAA mutates and is now TAG, hey, it's okay, because now I have AUC. It's still isoleucine. It's a silent mutation. That is called a silent point mutation. You can have a missense mutation, where now, instead of being CUU, now it's AUU. It's not a stop codon, but it's a different amino acid. It's no longer isoleucine, it's leucine. And if in a missense mutation, the R group was originally like polar and now switches to be non-polar, that's going to cause have consequences in how that protein folds up. But if it goes from like non-polar to non-polar, it should have like no effect on the folding up of that protein. So when the standard talks about the consequent phenotype, that's implying that you understand how mutations can be related to protein shape and therefore how they work. It's not always a bad thing. Mutations can also be beneficial. So here's a summary of the different types of mutations. There's a picture zoomed in. Errors in DNA replication or in DNA repair mechanisms and external factors, including radiation and reactive chemicals, can cause random changes or mutations in the DNA. So when you expose yourself to different chemicals, you can be inducing mutations. And yep. Okay. Errors in mitosis and meiosis can result in changes in phenotype. So that's like polyploidy in plants, or like in humans with Down syndrome. So they have like that extra chromosome. So non-disjunction can result in changes. Cute little babies. Okay. Oh, my shorts! What? The picture didn't have shorts originally. So, like here, this male has an extra Y chromosome, and the body type is slightly different, a little bit rounder, and has smaller testes. And so that's why I put shorts on, because we don't need a picture of that. And so, okay. And so then here you can see this sweet little girl is missing a second X chromosome. Now in females, we have 2 X's, but one of our X's condenses down to a bar body. It's all heterochromatin. What? And so it's random which X is expressed in each of our cells. But if you're born with just one X, you're only getting the genes that are on that one. And so like here you have consequences. So they have like a webbed neck structure. Changes in genotype may affect phenotypes. What? Changes in genotype may affect phenotypes that are subject to natural selection. That mutation could change your phenotype. That could increase your fitness, could decrease your fitness, or can make you neutral, but it depends on the environment. So here we have, I use antibiotics as our example. So genetic changes that enhance survival and reproduction can be selected by environmental conditions. So if you have a bacterium that is a mutant that has 2 mutations without antibiotics, that'd be like a neutral change. Kind of like, who cares? For driving this, by exposing ourselves to antibiotics on a daily basis. 75% of the antibiotics made in the United States are given to animals. So when you eat your dairy or meat products, you are exposing yourself to antibiotics driving the evolution of bacteria. What? Okay. The imperfect nature of gene replication or repair increases variation. So every time your cell divides, there's about 30 new mutations. Hopefully they're happening in your junk DNA areas and they're not affecting your phenotype. But if they happen in your gene, that can have an impact. Now, which kinds of mutations are going to be passed on to the next generation? Only the mutations that occur in gametes. If I get a mutation in my skin, like I'm going to go to my kid's soccer game after this and I'm sitting in the sun and I don't have sunscreen on, that's UV light that damages your DNA. It breaks the bonds and it causes thymines to pair with thymines. And so now if I go get UV damage, because it's an hour-long game, and I get mutations, it could be just skin cancer, and then I get pregnant. I'm not. But let's say I do. That skin cancer would not be passed on because that mutations did not happen in my eggs. It was only in my skin cells. Okay, so it has to, for mutations to be passed on to the next generation, that have to happen in your reproductive cells. Okay. Bacteria. We talked about this earlier. Bacteria. Wait. Let's summarize different. Prokaryotes and eukaryotes. We talk about variation. Variation is necessary for natural selection, and it kind of helps out evolution of life, right? And so by creating differences. So we look at eukaryotes, which are you and I. We're going to have, and plants and fungus and other eukaryotic cells. We have sexually reproducing populations. But you can also have mitosis with asexually reproducing. But let's go with sexually reproducing eukaryotes. With that, the three sources besides mutation of variation are crossing over, independent assortment, and random fertilization. Bacteria reproduce asexually by binary fission, making clones of themselves. They're made they have mutations that can create variation, but there are three ways that they can acquire variations. You have transformation. We talked about that with Griffith, where they pick up DNA from their environment. They have transduction, where a little phage virus accidentally transfers bacterial DNA and not virus DNA. And then you have conjugation things with the sex pillars, and it can exchange pieces of DNA. Okay, and so with this, there you go, so pretty. Okay. Phages, but then that DNA. All right. Oh, and then transposons. I never talked about. These are where DNA jumps. It's like jumping genes and actually will move to different areas, which is kind of different. So here's a transposon. It gets inserted into a piece of a different gene. So that can happen in bacteria too. So then here you can see here's a transposon. And so there's an enzyme that will actually cut this gene and move it into the disrupted gene. So that can also be variation. Okay, here, what is this? Transformation, right? You could have either just a piece of DNA that becomes incorporated into the main chromosome or a plasmid. Which one's this? Transduction, where the phages accidentally take up a piece of bacterial DNA. So you can see this little one. Oh, you messed up, buddy. And it's going to go and it's going to spread bacterial DNA, not virus DNA. Then you have in us, we've talked about this a few times already today, crossing over an independent assortment. Now, bioreplication differs from other strategies because it generates variation in various mechanisms. So with viral DNA, especially retroviruses are the ones that use reverse transcriptase. So as they start with RNA and use reverse transcriptase to build the complementary DNA sequence, they make mistakes. And now that is new variation in viruses. They mutate constantly, and that's why their antigens on the surface are constantly changing. And so vaccines and memory cells rely on those not changing. So if you have rapidly mutating viruses, it's really hard to make effective vaccines. It's not going to work in the traditional sense of attaching to antigens. Okay. So that reverse transcriptase, a lot of problems. We talk about viruses. Viruses have two different reproductive cycles. You have the lytic and lysogenic phases for a bacteria. Bacteriophage virus. So what's going to happen here is you're going to have the phage is going to infect a bacterium. If that DNA of the phage lays dormant, it hangs out and just lets the bacteria reproduce, that's the lysogenic cycle. And then if it receives some kind of environmental signal and becomes active, that's the lytic. And in the lytic cycle, it's going to multiply, and then bust out of the bacteria, killing the bacteria, but then they get to go and infect others. So you have both the lytic cycle and the lysogenic. And in the lysogenic, it just hangs out and stays dormant. Communication involves transduction of stimulatory or inhibitory signals from other cells, organisms, or the environment. So we talked about this in the nervous system. You have excitatory and you have inhibitory signals. Postsynaptic potentials can be either excitatory or inhibitory. If it's negatively charged ions, well, then that's going to be an inhibitory one because it brings you away from threshold. If it's an excitatory one, it's going to be positive ions because it's bringing you closer to threshold to have the message go. And then in single-celled organisms, we have quorum sensing. In quorum sensing is how bacteria communicate. So with quorum sensing, it relies on signal transduction pathways and turning on gene expression all at once to bring about their cell responses. And, oh, there you go. There's the ones on your teeth, yo. To make the capsules. That's so neat. Multicellular organisms, signal transduction pathways, coordinate activities within the cell. So with this one, you have, I already talked about epinephrine. So that would be an example of a signal transduction pathway. And it coordinates activities within the cell. So the organism can function as a whole. That standard is neat. Signal transduction pathways coordinate activities within cells that support the function of the whole organism. Epinephrine is a perfect example. Here's one cell receiving a signal, but then glucose is released into all of the body, so the whole organism can survive. When I first read that, like 5 or 6 years ago, I was like, what? What? Because, what? No, it makes perfect sense. Okay. So here's your epinephrine attaches to a G protein, and then you have signal gain peak gets activated, phosphorylation cascade, and then it activates the enzyme that will break down glycogen by hydrolysis into glucose, raising your blood sugar in the fight or flight response. What? Ooh, cells communicate by sub-cell contact. Ooh, plasmodesmata. That's for plant cells. It's little channels in the plant cell walls, so ions and stuff can move through the cytoplasm. Oh, look at all those plasmodesmata. What? Humans or like eukaryotes have animals have gap junctions. They're not as common, but we can share like a little channel between our cytoplasms. Oh, okay. Ooh, we have short distance. So short distance would be like paracrine signaling or local regulators. You see that in the histamine used in the inflammatory response, cytokines in like the humoral response, cell mediated response. We see this in neurotransmitters, etc. Like really close by things. And then so the growth factors would be for like the cell cycle, like telling cells in your body to divide. Signals released by one cell type can travel long distances to target cells. So that's going to be like your endocrine system with your hormones. They travel in your blood to your target cells. Oh, okay. So here's a summary, right? You have your ligand-gated ion channels, your G proteins, and your tyrosine kinases. So here, if you have a steroid hormone, it's going to travel into your cell, will attach to receptor side, turn on gene expression. And then, look at mRNA protein. And then you have the protein-based ones that will attach to a protein on the surface. Okay. This says conditions where transduction is blocked or defective can be bad. So the perfect example would be diabetes, because now something is like not being communicated, and so you have consequences. So diabetes is a perfect example of that. Remember type 1 versus type 2? Do you guys? So in type 1 is where the pancreas, the immune system attacks the pancreas and doesn't produce insulin. And then in type 2, it produces insulin, but for some reason, the protein receptor is not receiving the message. Okay, they become resistant to it. Organisms exchange information with each other in response to internal changes and external cues, which can change behavior. So it's like pheromones and mating season and breeding season. So the behavior is changed based on environmental cues like seasons. Oh, it's time to reproduce. Okay. So this is pheromone-induced ovulation in the non-breeding season. So even though it's non-breeding, they can have like pheromones. They're going to have that female ovulate. Fascinating. So we have a large pheromones can change your behavior. And then you have here the kin selection, where you have the warning call. I think that's what this is. Mean distance move from this is sorry. But do you remember the prairie dogs that get that warning? They live in groups, but the males don't stay because they end up mating with their sisters and their cousins. So the males move out of the area where the females stay to prevent like inbreeding. Here's some of the prairie dogs. Okay. And then you have communication occurs through various mechanisms. So this is like animals marking their territory. You have pheromones. Okay. And then communication is you have visual, auditory, tactile, and chemical. This is also big idea too. So communication tends to be a big theme in our year, because if you talk about like evolution, well, you tie in communication with natural selection. Talk about big idea too, with energy and communication is part of big idea 2. It's also part of big idea 3. So I feel like communication is a big thing. Oh, remember this one? Natural selection of poor whale. Okay. Okay. The neuron is a basic structure of the nervous system that reflects function. So action potentials propagate impulses along neurons. So this is where you have the different gates opening and closing with the different ions. When you get over to the axon terminal, you have your synapse. Your synapse is a gap between two neurons. So here you have little vesicles storing neurotransmitters. When the voltage changes in the axon as it's traveling down, when the change in voltage gets to the end, it's going to open the gates for calcium ions. The calcium ions are going to flow inside that causes little vesicles holding neurotransmitters to then move. And then by exocytosis, those neurotransmitters will be released into the synapse. And then those neurotransmitters are going to act as ligands for the next ligand-gated ion channels. So on this cell here, these gates will open and new ions will rush inside, depolarizing or hyperpolarizing the next membrane to lead to an excitatory or inhibitory postsynaptic potential. Okay. Oh, look, so pretty. And there's different types of amino acid. Yes, there are. But different types of neurotransmitters that can either be excitatory or inhibitory. And there's different parts of the brain. So if we look at like the three main parts, you have your forebrain, which is like the cerebrum in this area. You have your brain stem, which is like your midbrain, and then your hindbrain. So when we look at back here, your cerebellum is for coordination of movement, and then your brain stem is like the oldest part evolutionarily and deals a lot with homeostasis, like breathing and balance. Okay, that's the end of Big Idea 3. All right. It's 11:12. We ready to do Big Idea 4? It's about 97 slides. In like 40 minutes? Here we go, guys. Here we go. Do you want to stretch for like a second? Just stand up, get sugar to your brain, and then we're going to switch over to Big Idea 4. So many negative sounds. Do you hear that? Look how much you learned.
Oh my god.
I'm thinking if my parents said that I'm
I only thought to like look at that movie. Plus, even she was talking about
I have
animals.
no idea about any of this stuff. It's just like, yeah.
So what are you watching?
If they're not, I'm gonna watch it.
Hi, Tristan, if you're watching.
Okay, guys. What are I or wait, what's this one? Do you have stuff? It's extra credit, you know? All right. Here we go, guys. So structure and function of polymerase. Here we go. Our Big Idea 4, I feel like, is like random mix of stuff. So the way it's going to work is we're going to look at macromolecules, and then we're going to go into, I think enzymes are next, and then cell organelles, and then ecology. So it's like a random mix of things. So ecology kind of ties into Big Idea 2 and 4 here. So structure and function of polymers are derived from the way their monomers are assembled. So if you look at glucose, let me start over. If you look at starch versus cellulose, they're both in plants, they're both polysaccharides, and they're both made of glucose. But the way they're assembled is different. So, in cellulose, that's where you have the alpha and beta forms of glucose, like alternating. And then in starch, it's all the alpha forms. So they're both polysaccharides, but they have completely different functions. And so with this, so the function of them, so the starch is going to be energy storage for the plant, like a potato is a whole bunch of starch. So when you eat that potato and you hydrolyze, you break down that starch into glucose, you have lots of sugar. And I know when I eat potatoes, like French fries, before a workout, I have a fantastic workout. I'm faster, I'm quicker, like I can last longer in my running and stuff. And so it's all due to that high. Probably shouldn't have French fries on the way to a workout. But you know what? That's life. And so anyway, but cellulose, I'm not going to go have a salad before a workout because cellulose is what makes plant cell walls. And even though it's all glucose, I can't access it. That's not available free energy to me. So it wouldn't make sense. Oh, I have a big race. I'm going to go have a big salad. That's lots of stuff. No, I'm going to like chew it up and poop it out. And so the starch, I will hydrolyze and get that glucose. And then you have the way their monomers are assembled. I think that's also might tie into proteins, right? All the different amino acids, like the order of that primary structure will affect how it folds up. And so when we build polymers from monomers, we're relying on dehydration synthesis, removing of the water. We also have hydrolysis, where you're going to break the bonds down. We do that a lot when we digest our food. We do hydrolysis to break things. Our lysosomes also have the enzymes. They're going to do hydrolysis to break down organelles. Now, the cool thing, guys, in biology is when they talk about molecules or macromolecules, everything is either going to be a carb, lipid, protein, or nucleic acid. And that's what these things are, macromolecules. So here, when we look at them, our 4 macromolecules, we have polypeptides form proteins. So proteins are macromolecule. We have our DNA. DNA is made of nucleotides, over and over and over, a long chain of nucleotides. That's going to be our a polymer. And then you have your carbohydrates. So a long chain of glucoses will be a polysaccharide. Now, the one that doesn't fit in the monomer polymer style is fats. So here I have a triglyceride. It's made of a glycerol with 3 fat acids. So it's still a large molecule made of C, H, and O, but it's not a polymer like the other 3. It's not made of repeating units. Okay. So here we have starch. It's alpha, 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 alpha. Here we have cellulose. Alpha, beta, alpha, beta, alpha, beta. Okay, okay. And then they form by dehydration synthesis for moving on water. And so when we look at carbohydrates, you have glycogen. Glycogen for us is branched. That make it easy to break off different branches because we're going to store glycogen in our liver for energy storage. And so we're going to break off pieces. Like I haven't eaten yet today, but look at how much energy I'm using. So right now you can bet that my hormone called glucagon is going to my liver and it's telling my body to release sugar to maintain homeostasis so I can keep teaching you at a high energy or a high energy level or something, right? And so here we go. So when we look at this, here you have your DNA is a polymer. It's a macromolecule, and then it has the codes for proteins. So I just think that's kind of a neat picture. And we also have carbohydrates. This here has 3 of the 4 polymers. You have your cell membrane made of lipids. Then you have, this is a protein you can't really see because the picture's bad. And then you have your carbohydrate chains coming off, right? But what? Okay. So you are what you eat. So we eat these things, carbs, lipids, and proteins, and then we break them down. We hydrolyze them, and then we use those monomers to build our cells. Okay, okay. So eat well. Eat well. Make good choices in life. Or, right, where else? And so proteins, carbs, and lipids, though, and then we can break them down for either building our cells in biosynthesis or for energy production. They're all made of CHO. And then some of them have N, protein, and DNA. And then DNA has P as well. But what? Okay. Please don't miss that. So amino acids. If you look at amino acids, wouldn't that be so bad if that was like part of one of the essays? And like CHOMP? Anyway, so now here you have amino acids. They form peptide bonds. There's different R groups coming off, but each amino acid has the same like major parts. You have your amino group and your carboxyl group. And it's the carboxyl group as what attaches to the amino group to form that peptide bond. Then as it folds up, you have your primary structure is the order of the amino acids, and then it starts to do an alpha helix or a beta pleated sheet, which are going to be based on hydrogen bonds. Then you have your tertiary structure, which is going to rely on like disulfide bridges, nonpolar interactions, ionic bonds. And then you'll have that fold up. And then if you take multiple polypeptides together, that's the quaternary structure. Hey, this is hemoglobin that we talked about in class the other day with its affinity for oxygen. And when it gets to be like a higher temperature or like a lower pH, it like lowers its affinity for oxygen and lets it go. Let it go. And then you have, this is just looking at proteins in the body. So proteins are made by transcription and translation. And there's different functions. We have them in our cell membranes. We have them, our antibodies, so part of our immune system is a protein. We have hemoglobin to carry oxygen is a protein. We have protein receptors on our cell membranes. Our muscles are proteins. Our hair, keratin is a protein. Like our pigment is a protein. Like your phenotype is based on proteins. And that protein is based on your genes. And then your genes are determined by like evolution, really, like a natural selection and mutations being selected for or against. Oh, okay. Oh, for the love of lipids. All right, so when we look at lipids have so much stored potential energy because of all these bonds. All of these bonds can be broken, and the electrons can be used to produce electron carriers and carried over to the electron transport chain. But then they can be oxidized for the oxidative phosphorylation to make ATP. There's so much energy in lipids. Okay. So that's why like seeds, like little tiny baby seeds, like a sunflower seed is like a mama plant doing photosynthesis, making G3P, and then like turning into lipids to store as much energy as possible in the smallest place. And so when that seed germinates, it can use all that stored energy to build its roots, to grow its roots by mitosis. And then like the plant, once it like builds its shoots and then its leaves, and then it will rely on the sun for energy. But initially it's going to rely on energy and cellular respiration for its roots during germination. It's not until it has leaves that will rely on the sun. Okay. And that's why seeds are so fattening, because that mama plant's really trying to get them to survive. So lipids can be based off of, like, a lot of times, like cholesterol is a lipid, and that's like a precursor for building other molecules. But here you can see, too, we have some fat-soluble vitamins that will be stored in your fat. They also form your cell membranes. And they act as like a waterproofing. So in ducks, and birds, it can like waterproof their feathers with an oil, but also plants. Plants have a waxy cuticle for waterproofing on their surface. And so here's a feather. You can see the water beating up on its surface. And so that waxy cuticle is on the surface. Underneath is where you have the guard cells with the stomata for the gas exchange in plants. We use all of these for energy. Now, fat is not water soluble. So when you eat fat, so let's say you have like chicken tenders or French fries, like, I don't know, a donut, bacon. You have something that's like fatty. Fat and water don't mix. So when you eat that, here it is coming in from your stomach. The fat is going to repel the water in your intestines and kind of clump together. So what will happen to break this down, though, we need to increase the surface area. So you have bile. If you've ever thrown up and you're like dry heaving, have the flu or something, and you're throwing up, and it's green, like that bile. Do you know what I'm talking about? It's like green or yellow. At the end, there's no more food left. That bile is stored in your gallbladder. It's made by your liver. And so what will happen is when you eat a meal, bile is dropped into your small intestines. Bile is going to then mix with the fat and kind of break up the fat, like make it spread out. And now your enzymes can get in there and hydrolyze it and break it into smaller pieces. It's kind of like if you cook bacon and you go to wash the pan and you use just water, the water and bacon grease don't mix. But if you add soap, the soap kind of breaks up that fat and now it can wash away. The bile is like soap. It like breaks up, like spreads out your fat and lets enzymes like break it apart. So people who have like their gallbladder removed, they don't have a way to store bile, so they're going to need to like watch how much fat they take in at a time. The other thing is that the bile is made from cholesterol. Bile does not mix with water either. So for a person to lower their cholesterol, they want to try and figure out a way to get bile out of their body. So to make new bile, you'd have to use your cholesterol to build new bile. So when you see like those commercials about Cheerios, oh, heart health, lowers your cholesterol. Cheerios, oatmeal, soluble fiber, that bile, when it's released, is going to get stuck kind of in that oatmeal or those Cheerios, and then it's going to be removed from the body. The body needs to use cholesterol to make new bile. So therefore, your cholesterol levels will get lower after eating oatmeal every day for breakfast, or lentils, or any other kind of soluble fiber. Otherwise, without that soluble fiber, the bile just gets reabsorbed and reused. So that's how like you can choose certain foods to lower your cholesterol. You can also choose foods to increase your cholesterol, like steak, I don't know, beef, pork have high cholesterol. Okay. Anyway, I decided to let you know that's a little bit of, if you ever find yourself with high cholesterol in life, like at dangerous levels, try and increase your soluble fiber because it'll act like a sponge, take out the bile, and you have to use cholesterol to make new stuff. Okay, so here you have the fat drops or whatever, and then the bile will help to break it down, so that way the it's called emulsify, and now the enzymes can access it and break it into glycerol and fatty acids. We just talked a whole bunch about DNA and RNA, so I'm going to skip that polypeptide. Is that okay? I'm probably not polypeptide, polymer. And now the directionality influences structure and function of the polymer. So how you build it matters. So like structure and function, starch and cellulose, how you build it matters. Same thing with proteins. How you build it matters. Depending on the order of the R groups will change the structure of the protein. Okay, so now we move into organelles. Organelles, I'm going to go kind of fast. Is that okay? Ribosomes. Ribosomes make proteins. Done. I'm just kidding. We're like kind of done. And so let's talk about the two different places you find ribosomes. You can have a ribosome that's located on the rough yard. If a ribosome is located on the rough yard, it's going to make a protein that will end up leaving the cell. Or it'll make a protein that will become part of the cell membrane. It's going to go through the endomembrane system from really starts, you have the nucleus. The nucleus has the DNA that has the code to make the RNA. The RNA will be edited in the nucleus. It's going to go to the rough yard where there's a ribosome. The ribosome will then read that mRNA. It's going to make a protein. That protein will then get carried in a vesicle to the Golgi. The Golgi is going to package, modify, and sort. And then the vesicle will carry it to cell membrane. It's going to be sent out by exocytosis or become incorporated into the cell membrane. However, there's some proteins that don't leave our body. I mean, yeah, true. Some proteins that don't leave our cells, like protein kinases. A protein kinase is an enzyme we need within our cell. So those proteins will be made by ribosomes that are free-floating ribosomes. You have free ribosomes floating in your cytoplasm that are going to make proteins that stay within your cell. And then you're going to have ribosomes that are attached to the Reff-ER. They're going to make proteins that are going to leave your cell or become part of the cell membrane. Those are called bound ribosomes. So you have free ribosomes and you have bound ribosomes. And where their proteins end up really is where they're made, like where the protein is made. Does that make sense? Like which ribosome? Okay. So protein synthesis. Oh, there's different sizes of ribosomes. So us eukaryotes and prokaryotes have different size ribosomes. So some antibiotics will actually target bacterial ribosomes, and ours are safe because they're slightly different subunits at different sizes. So that's what these are, different measurements of the subunit and sizes of it. So prokaryotes also have ribosomes, and we do too, but we have different sizes. And that's why if you have an infection, an antibiotic can target theirs and not yours. Okay, ribosomes, Joe. Ooh, mitochondria have ribosomes as well. Mitochondria have their own ribosomes. They have their own DNA. They have their own proteins. They're produced by binary fission. What? Evolution. Okay. Then you have two forms of ER. You have the Reff-ER and you have the smooth ER. The Reff-ER has ribosomes, and that's why it looks rough, and it's involved in protein synthesis. The smooth ER is smooth like butter. Butter is a lipid. The smooth ER makes lipids. And so they also detoxify the body. So if a person is like drinking alcohol or something, the smooth ER will help to break it down. So a person who drinks a lot of alcohol throughout their life will get more and more smooth ER inside their liver cells. And that's how they build up a tolerance. So there you go. Okay. And they also detoxify drugs and poisons in your liver. All right. Then we have the Golgi. The Golgi is a membrane-bound structure that consists of a series of flattened sacs and membranes. Now, cool thing about the Golgi, so yes, it's involved in like the proteins being like modified and getting sent out of the cell, but your lysosomes actually are made from your Golgi. When you see these like budding off the vesicles, like here, this is going to become your lysosome. And your lysosome has proteins inside, has hydrolytic enzymes. Inside your lysosome has a pH of 5. It's a more acidic environment, so those enzymes only work best inside that lysosome. So they leak out into the cytoplasm where you okay, let me start over. Actually, I think there's a slide on lysosomes. We'll get there, but can I talk about lysosomes now? So your lysosomes are your organelles that are going to hydrolyze and break things down. We saw this in the immune system. When a macrophage engulfs like a pathogen or a microbe, your lysosomes come and they fuse with it and digest. But if you have old mitochondria, your lysosomes will break it down. If you're an amoeba that's engulfing its food, the lysosome is going to use indigestion of food. So your lysosome has hydrolytic enzymes that digest and break bonds by hydrolysis. This is also where those people with taste saps out of your lysosome, they run the risk of hydrolyzing the organelles and the things you need. So the cool thing that has evolved is that the pH of inside the lysosome is a pH of 5. And so if they do leak out, those enzymes become denatured and they lose their shape and now they don't work and your organelles are safe. I think the lysosome is my favorite organelle. Who is a favorite? I do. Okay. Then you have mitochondria, specialize in energy, capture, and transformation. I think we've spent a lot of time kind of outside the respiration. And so we see like on transport chain. Isoleucine. Hydrolytic enzymes. What? Look at that food vacuole. Oh, man. Okay. Oh, you can break down damaged organelles. It can be used for like food particles for an amoeba. Anyway, I just really like it. Vacuoles are used for storage. Vacuoles are used for storage in plants. And they can help with intracellular digestion and storing waste products for plants. And they can store pigments and poisonous substances. And they also help with the plant. When the vacuole fills up with water, it presses on the cell walls and the plant stands straight up. Okay. Oh, there you go. Hypotonic solution. Plants prefer hypotonic solutions, so the water flows in by osmosis and then it fills up, and now they're standing upright. Because not all plants have bark. Look, a tree has tree bark. That's like dead plant cells. Actually, bark's more complicated, but it's like dead tree cells that give it structure. But like, think about a dandelion or like some little tiny flower that doesn't have a bark. It's going to rely on standing straight up, or like basil plants, because the back are going to be full. That's when you don't water your plants, so they're kind of wilted. But when you water it, they puff back. They don't puff back up. Osmosis happens. Okay. So there's your back. Oh, chloroplasts, specialized for energy capture. Okay. Do we need photosynthesis right now? No. Then we have differentiate. Oh my gosh, now we're switching gears totally. Differentiation in development.